There's a mystery unfolding right now in the world's oceans. The victim? Billions and billions of tiny shrimp. The crime scene? Well, it's aquaculture farms all over the globe. And the culprit? It's an invisible killer that leaves its victims pale, empty, and ghost-like. So let's dive into this investigation because this isn't just a fish story, it's a potential pandemic threatening one of our major food sources.
Our story starts with a deadly disease that, for a while, seemed to be contained to just one country. But now, for the very first time, it has officially crossed a border, and that has sounded a massive alarm for the multi-billion-dollar global shrimp industry.
And right here, this is our central clue. Take a look at the shrimp on the left. See how it has that dark line running down its back? That's a healthy, full digestive system. Now look at the one on the right. It's almost completely see-through. This is why they call it translucent post-larvae disease, or TPD. It's not just a change in color, it's a sign of a really rapid and really fatal infection.
So let's look at the timeline of this thing. It first blew up in China back in 2020, and it absolutely wiped out a huge number of shrimp hatcheries. For 3 years, it seemed like they had it contained. But then, late in 2023, a call comes in from a completely different country in Southeast Asia. The report is grim, huge, sudden die-offs. And pretty soon, scientists confirmed everyone's worst fears. TPD had officially gone international.
And when I say the die-offs were huge, I am not kidding. At this new location, more than 70% of the young shrimp were dead in less than a week, just 5 days. The speed of this thing is what's so incredibly terrifying. It was crystal clear that investigators had to find the killer, and they had to find it fast.
And that brings us to the forensics part of our story. Samples from the crime scene were rushed to the Aquaculture Pathology Laboratory at the University of Arizona. Their mission? To hunt down, isolate, and identify the pathogen that was causing all this carnage.
So, after digging through the samples, they narrowed it down to 2 main types of bacteria they kept finding on the victims. You can think of this as a microscopic police lineup. On your left, we have suspect number 1, which forms these really distinctive green colonies in the petri dish. And on the right, suspect number 2, forming those yellow colonies.
And the lab work put some names to these faces. Suspect 1, our green one, is a strain of a bacteria called Vibrio parahaemolyticus. Suspect 2, the yellow one, is Vibrio alginolyticus. Now, both of these belong to the Vibrio family, which is a group that's pretty notorious for having some nasty characters in the marine world.
So now we have our 2 prime suspects. But here's the million-dollar question. Are they working together, or is one of them just a harmless bystander and the other a super virulent killer? To figure that out, the scientists had to go deeper. They had to look at their DNA.
Which brings us to the next phase, DNA fingerprinting. The investigators are now basically searching for specific genes that act like genetic weapons, the very weapons they know the TPD killer uses. And wow, the results here could not be clearer. The scientists checked for 2 different sets of toxin genes. First, a set related to a totally different shrimp disease. And nope, nothing for either suspect. You see those red X's. But then, when they looked for the specific TPD toxin genes, they got a perfect hit. Only suspect AG1, our green colony bacteria, was carrying the genetic murder weapons, a big green checkmark.
And here it is, the raw data, the DNA evidence itself. This is a technique called gel electrophoresis, but you can just think of it as a DNA fingerprint. And you see those bright glowing bands in lanes 8, 12, and 16? That's the smoking gun. It's a perfect match, confirming suspect AG1 has the TPD genes. Now look at the lanes for suspect AY7. Totally empty. The DNA doesn't lie.
So, the genetic evidence is absolutely conclusive. Suspect AG1, that Vibrio parahaemolyticus strain, is our prime culprit. But, in science, you have to prove it beyond a shadow of a doubt.
So that means it's time for the final test. This is basically the scientific equivalent of recreating the crime scene. To be 100% sure, the scientists had to take their prime suspect and see if it could cause the exact same disease in perfectly healthy shrimp.
The experiment is called an immersion challenge, and it's pretty straightforward. Step 1, you take healthy 15-day-old shrimp and you put them in water with each of the bacterial suspects. Step 2, you just watch them for 4 days. And step 3, and this is the key, to prove it's guilty, the bacteria has to cause the exact same translucent symptoms And they have to be able to pull that same bacteria from the new victims.
And the results? They were just staggering. The control group, with no bacteria, 0 deaths, no surprise there. Suspect AY7, the yellow one, basically harmless. But suspect AG1, oh, it was absolutely devastating. At high doses, it killed 100% of the shrimp. 100%. Case closed.
This scientific jargon here basically tells us how deadly this thing is. It's a measure called the LC50, which is just the dose needed to kill half the population. And for this bacteria, that number is incredibly low. To put it simply, it takes a shockingly small amount of this pathogen to cause a complete catastrophe. And if you want to see what that looks like up close, check out these microscopic crime scene photos. On the left, that's the complex, organized tissue of a healthy shrimp's main digestive organ. Now look at the picture on the right. That is what AG1 leaves behind. It's just total cellular destruction. It literally dissolves the animal from the inside out. Now, just when you think the whole case is wrapped up, neat and tidy, the scientists discovered something they really didn't expect, a twist in the story.
They decided to run the exact same experiment, but this time they used slightly older shrimp, 30 days old instead of 15. And would you look at this? The mortality rate absolutely plummeted. Even with the highest dose of that same deadly bacteria, only 20% of the older shrimp died. It seems that as the shrimp get just a little bit older, they develop a really strong natural resistance to this killer disease.
So, the specific case is solved. We have our killer. It's the AG1 strain of Vibrio parahaemolyticus, and we know it's a specialist that targets the youngest, most vulnerable shrimp. But solving this one mystery has just revealed a much, much bigger problem.
This event officially puts TPD into a very serious category. It's now a transboundary disease. That's the official term for a pathogen that can move fast across borders, whether it's through shipping, water currents, you name it. And these are the kinds of diseases that can trigger major international crises, messing with food security and entire economies. And get this, our killer is already a pretty tough opponent. This testing showed it's completely resistant to 2 of the most common antibiotics out there, penicillin and ampicillin. Now, thankfully, it's still vulnerable to a bunch of other drugs, but this is a really worrying sign that it's already evolving and developing resistance.
So what this all boils down to is a massive, urgent warning. Finding TPD outside of China for the first time is a huge wake-up call for the entire world. The only way we can fight this is with non-stop surveillance, really strict biosecurity measures on farms, and tests that can catch an outbreak before it has a chance to explode.
You know, the case of the translucent shrimp is a pretty powerful reminder of just how fragile our global food systems can be. We caught this one early, which is good, but it really leaves us with a critical question. In a world this interconnected, what's the next microscopic threat that's silently crossing borders, and are we going to be ready for it?